Previous lesson, like I told you, we talked about how the rate of respiration is controlled, or factors that do affect the rate of respiration. Where we saw on a cellular level, and we saw that respiration is controlled by negative feedback mechanism, and then on the whole organism level, for the whole organism as it is, then respiration is controlled by factors, the three factors: the cell, the organ, the size of an organism, and then the state of activity of an organism, and temperature. So we talked about temperature in our previous lesson. You can go there and check. I have the recorded video, it's there. And we saw that respiration increases with increase in temperature, and we try to explain all this in poikilotherms, in homoiotherms, how temperature really affects and how these organisms cope up with temperature as far as respiration is concerned. So today let's talk about other factors such as body size. And we are aware, actually I usually see this. It is called, like right now, it's raining right now, or it is threatening to rain. You see some people putting on jumpers, jackets, stuff like that. But others are just in their armlets. Examples for the case of ladies. For ladies, you find a fat one in just an armlet, but a tiny one looking for sweaters, jumpers, the coats from the boyfriends, all that. They do respond to the changes in the temperatures avoided. But what is the effect of body size to rate of metabolism vis-a-vis temperature loss? Okay, if you can analyze that, if you can understand that, then you know how body size really affects the rate of respiration. And simply, large organisms, they have low rate of respiration, and small organisms have a large rate of respiration. And that's what I expect from you. Okay, if I set a question and say, how does body size affect the rate of respiration, and you start answering by saying small organisms with large surface area lose, and I'll be like, man, what is this? What are you trying to explain? Okay, so the first thing is, first tell us how, and then after the how, then you give the why. But don't start by answering why when we don't know the how. Okay, so body size, small organisms have a high rate of respiration, and large organisms have a low rate of respiration. And it is simple. Small organisms do have a large surface area to volume ratio. And I know you've ever calculated this, but we can do it again and see. So if we have two cubes, and the first cube, maybe it's 1 centimeter, and all sides are equal, regardless of me drawing it in this format. All sides are equal. Then you have the second cube. Let's see the concept of surface area to volume ratio. You have the second cube, and this one. Oh my god, sorry for that. We have a second cube, and this one is maybe 2 centimeters. Okay? Oh my god. So this is 2 centimeters, and all sides are. Now if you look for surface area, okay, to volume, this ratio of surface area to volume, you find that the small cube, the ratio is high compared to a larger cube. Let's see how. So if the surface area is going to be area of one surface, okay, maybe let me use some different pen. People here prefer pink. So we shall get area, that is Katogo. So we shall get area of one surface, okay? And what is the area of one surface? It is side squared, which is 1 times 1, which is 1 centimeter cubed. Times the total number of surfaces, and the total number of surfaces, there are 6. There is this surface, there is this surface, there is this surface, there is the other surface down, okay, there is this surface here, and there is the other surface that is coming this side, okay, behind, and then there is this surface down. So those are 6 surfaces. So the surface area is going to be 6 centimeters squared. But the volume, it's 1 times 1 times 1, which is 1. So surface area to volume ratio, therefore it becomes 6 out of 1, which is 6. So the ratio here is 6. But for this case, the surface area is going to be 2 squared times the 6, which is 24. Is that okay? Centimeter squared. And the volume is 2 times 2 times 2, which is 80. So our ratio is going to be 24 divided by 80. And what is the answer? The answer is 3. So do you see that the ratio in large organisms, the surface area to volume ratio in large organisms is small compared to small organisms? So as size increases, then the surface area to volume ratio decreases. And so if the surface area to volume ratio decreases, then what does that mean? Large organisms lose heat much slower, or at a slow rate, than small organisms. So as a result, the small organisms, since they lose heat at a faster rate, they need to increase their respiration rate or their metabolic rate in order to generate heat that is lost to the surrounding. Hence, a higher rate of respiration. So, if this concept is key, it works best in both homeostasis, almost everywhere. Transport, talk about respiratory system. Like, please explain why some organisms require a specialized transport system, whereas others don't. So this concept is key. Then let's talk about other factor. And the other factor here is level of activity. And animals that are really active, they have a high rate of respiration. This is, anyway, we don't take things to be obvious. That's good. If you want to become a good teacher, don't take things to be obvious. So level of activity, it shows how active an organism is. Okay? For example, animals with a different physical state, okay, they do have a high level of activity. For example, you can consider the flying and then the sprinting. This, the Usain Bolt. The Bolt is really active. The flying birds, they are really active. So the rate of respiration in such animals is high compared to the rest. Then growth, organisms that are in their growth spurts, they have a high rate of respiration. For example, if you consider germinating seeds and then young ones in their growth spurts, Okay, they really do have a high rate of respiration. Then dormancy. Now organisms that are dormant, you know, we have what we call hibernation and estivation. These are state of physiological inactivity, if I'm to describe it as that, where an organism cuts off most of the body metabolism in order to serve, so in order to conserve the little food that is available until when factors become normal again, until when factors become favorable again. So organisms that are under hibernation, or organisms that are under estivation, or even seeds that are dormant, that are not growing, germinating, they do have a low metabolic rate, a low rate of respiration. So let's talk about RQ, or respiratory quotient, and see what it is. Now, it is the ratio of the volume of carbon dioxide that is produced to the volume of oxygen that is used in the same period of respiration. In the same period of respiration. For example, you consider, and okay, I will say that you supply oxygen, and maybe you've supplied 6 liters or 6 whatever decimeters cubed, you know how gases are measured, and then you produce carbon dioxide, and the carbon dioxide is lower. Because if we write this equation, let's first write it. If we write this equation, carbon dioxide, okay, glucose, H6, H12, O6 plus 6 oxygen, 6, we shall produce 6 carbon dioxide from our equation plus the water. And how much water? 6 water molecules plus energy. Now, ideally, if we are to consider an organism that is really undergoing aerobic respiration at normal condition, the RQ of this person is the ratio of carbon dioxide that is produced to the ratio of oxygen that is consumed. And using volume ratios, we are seeing that this person is consuming 6 volumes of carbon dioxide. Okay, so it is consuming 6 volumes, is producing 6 volumes of carbon dioxide, and this person is consuming 6 volumes of oxygen. So the RQ is 1. Okay, the ratio of carbon dioxide produced to oxygen consumed is 1. So basing on that ratio, any deviation, we can use it to tell. Any deviation from this ratio, we can use it to tell why there is a deviation. For instance, if the organism is under anaerobic respiration, is this ratio still going to be 1? Really? Think about it. Is this ratio going to be 1, or below 1, or above 1? So think about it. Number 2, for plants, where carbon dioxide produced can be used in photosynthesis. So at a high rate of photosynthesis, is this ratio going to be still 1? Is it still going to be 1? If no, then why? So basing on the RQ values, okay, then we can use these RQ values to tell the state of activity of an organism, to tell which type of respiration this organism is undergoing, and so on, and so on, so on. Okay, yeah, for example, you can check. So this is glucose, then this is oxygen, this is carbon dioxide that is produced, and this is water. And the RQ is 6 volumes of carbon dioxide divided by the 6 volumes of oxygen, then we shall get 1. So here our RQ, our respiratory quotient, is 1. Okay? Now, is the RQ important? Are these RQ values really of any biological importance? And I can say yes. And if you want to confirm, is a question I posted in our class. If you are keen enough, then you have seen the question. Then if you haven't, still it's okay. You will see it. Okay? You see the question and enjoy it. Make sure you do it. So we can use the RQ values, okay, to indicate the kind of substrate that is being metabolized, that is being respired. Whether you are respiring carbohydrates, whether you are respiring proteins, that is, we shall know that either these organs may starve. Because for carbohydrates, you're supposed to get a ratio of 1 to 1. So if you get a ratio that is above 1, or a ratio that is below 1, then you are not respiring carbohydrates. And if you are respiring, then you are undergoing anaerobic respiration. So we can use RQ to indicate if the rate of respiration is undergoing, if it is aerobic or anaerobic. Whereby at low RQ values, that if the value is less than 1, okay, then all the carbon dioxide that is used, or some carbon dioxide that is produced, is going to be used in photosynthesis. So that now the ratio carbon dioxide out of oxygen is low. Okay? But if the rate of respiration maybe is anaerobic, then few or no oxygen, little or no oxygen is being used. Okay? And so the carbon dioxide that is produced is going to be high compared to the oxygen that is used. So the RQ value is going to be high. So now the question is, how shall we measure the volumes? Okay? How shall we measure the volumes of carbon dioxide and the volumes of oxygen so that we can determine the RQ values? Then we shall use a respirometer, or you can say a spirometer, which detects changes in the pressure, in the gas pressure. So basing on the changes, you can use it to determine the volume of oxygen and the volume of carbon dioxide so that you can determine the RQ value. So you can check on that question, please, and have fun and enjoy. The question is really interesting. Is that so? Any question? So that was basically what RQ is about. Okay? Yes, now let's do some few experiments about alcoholic fermentation in yeast. And these experiments, they are good. They can be asked. For example, UNEP 2019, paper 2 number 1. It was this. So you need to have a good insight, okay, as far as alcoholic fermentation is concerned. So this yeast, it's a fungus, okay, It is fungus. I think is it under ascomycetes, something of that kind. You can revise your classification, the classification of fungi. So it is fungi and it's a unicellular fungus, so unique from others. No other fungus is unicellular, and it strictly undergoes anaerobic respiration. And the product of anaerobic respiration of yeast is alcohol. And actually, industries have really used this yeast to make money. Even not only industries, even here at home. If you know you make bushera, eh? You've used this yeast and you have used this alcoholic fermentation so much. Okay? Yes. So the type of sugar that is best. So one of the experiments we can use the experiment to determine which type of sugar is best metabolized by yeast. Okay? That's one experiment. And we shall see. So we can determine which type of sugar can be best metabolized by yeast. And actually, for those of you who did senior 4 last year, this was your number. This was practical work. Okay? It was also about this. Actually, even the senior 6 you named, the numbers were the same. However, the complexity. Also, the number was about this. So when we are studying this, I usually give it enough attention because it can appear in both theory and practical. So please take care. So we can use to determine which type of sugar can be best metabolized. And the sugars you might have maltose, and this one maltose, it's a disaccharide that is made up of two glucose molecules. Okay? And it is formed from hydrolysis of starch, or partial hydrolysis of starch to maltose. Anyway, hydrolysis of starch to maltose. By which enzyme? By amylase. Amylase. So that implies that in yeast you have this enzyme, amylase, or ptyrin is diastase, I think. Not ptyrin, it is the same as salivary amylase, but in this case, let's call it, you can refer to it as diastase or amylase. Then we have glucose. Okay, yeah, we all know glucose. This is a monosaccharide. Okay, made up of 6 carbon atoms, which is C6H12, and O6. So basically, this is your glucose. Then we have fructose. This one is also a disaccharide, but it was a slip of tongue. It's a monosaccharide, but the good thing with this fructose, it can be converted to glucose and from glucose to this at any time. Then we have sucrose. Now this one is a disaccharide that is made up of both glucose and fructose. And then galactose, it is also a a monosaccharide that has almost the same structure as glucose, but the difference is on carbon number 2. If not 2, it is carbon number 4. One of the two. Just I need to go and check. Okay, where in one the OH is oriented outwards. Just you can check. Then lactose, this is a disaccharide that is made up of two monosaccharides, that is galactose and glucose. So you can carry out an experiment to determine which sugar is best metabolized, and basing on the data that is given, you can be able to conclude. Then you can also carry out an experiment to determine the effect of yeast fermentation on polysaccharides. Okay, so all these, they were disaccharides and monosaccharides. But you can also determine the effect of yeast fermentation on polysaccharides, such as starch, okay, starch and cellulose. So of the two, between starch and cellulose, uh-huh, which one is best metabolized, or what happens during their metabolism, their respiration? So basically that's what we shall be talking about as far as these experiments are concerned. But before you talk about experiments, let's first see some few methods that can be employed in order to study the respiration, alcohol, the anaerobic respiration in yeast. And the first one is, oh my god, we can use a gas pressure sensor, okay? Gas, gas pressure sensor, like this, gas pressure sensor. So this one, it just detects changes in gas pressure. As simple as that. Changes in gas pressure. Okay? So we can use this. Oh, it's okay. So like I told you so about that. So we can use a gas pressure sensor. Okay? So like its name, it is gas pressure sensor. It's a sensor that detects changes in gas pressure. Okay? Now, we all know that in respiration, carbon dioxide is produced. Okay? But since this is anaerobic respiration, okay? This is anaerobic respiration where there is no oxygen that is consumed at all. Okay? So if you carry out this reaction, sorry for that, if you carry out this reaction within a closed system, okay, where no oxygen enters, neither carbon dioxide moves out, it's a closed system. The carbon dioxide that is produced within respiration causes changes in pressure, okay, of this gas, so of the whole system. You are producing carbon dioxide, but you are not using any oxygen. After all, even the system does not have oxygen. Okay? So there will be a change in pressure. There will be an increase in gas pressure. But if it was aerobic, you remember, where you use 6 volumes of oxygen and produce 6 volumes of carbon dioxide. Okay? Then here, there won't be any change because what is used is replaced by the carbon dioxide. But in this case, you are using the carbon dioxide but no oxygen. So you are producing the carbon dioxide but no oxygen is used. So as a result, there will be a change. There will be an increase in gas pressure. And so this gas pressure sensor here, it will be the one to detect that increase in pressure. And so basing on how the pressure is increasing, okay, then you can determine, basing on how you are producing carbon dioxide, if you produce less carbon dioxide, then less pressure is detected. If you produce much carbon dioxide, then much pressure is detected. So basing on this, you can do the comparison between the sugars and you determine which one is best fermented. Okay? So basically that's the concept. So with aerobic, no change in gas pressure in the test tube. So rate of carbon dioxide is an indication of the rate of breakdown of the sugars. And we have an example here. Now we shall not dissect the whole of this question. These are full questions, but I just wanted to illustrate. Okay? Okay, so now in this question we are comparing, okay, between these two, between these sugars, which sugars is best metabolized, okay, and you can base on the volumes of carbon dioxide that are produced to tell which sugar is really best metabolized by the yeast. So the graphs, these graphs, the graphs of carbon dioxide evolved with time during respiration in yeast in different sugar solutions. And we are using a gas pressure sensor, okay, to monitor anaerobic fermentation as carbon dioxide produced causes changes in pressure in a closed tube. Okay, and this is a graph that we are getting. So you can see this is carbon dioxide. This is the volume of carbon dioxide that is produced. Okay, and the volume is assumed to be proportional to the pressure that is observed within the gas pressure sensor. And basing on this volume, you can tell that after 25 minutes, is it in minutes? Yeah, 25 minutes, you see where this one is. 50, you see where this one is. 70, you can see. 100, you can see. After 125 minutes, you can see. So basing on the volumes of carbon dioxide that are really produced in each, okay, that are really produced in each, then you can tell which sugar is best metabolized by the yeast. For example, you can see for maltose, you can check on the trend. So rapid. And the highest, I can say compare the trend in maltose and maybe sucrose because they are all disaccharides. So I can say compare the trend in maltose and sucrose. So you should be in position to compare. And with comparing, you give differences, and similarities. And please, giving differences here, don't put them in a table form.
Teacher,
Yes.
teacher,
Yes?
teacher.
Yes, or team?
You're glitching a lot.
Seriously? So somebody has requested to repeat the slide, which is okay. Which is okay. So like I told you, we can use a gas pressure sensor to determine of these two different types of sugars, which sugar can be best metabolized. If you are unmute, please, you can mute, it's okay. Which sugar can be best metabolized? And you know we have different sugars. We have the disaccharides, monosaccharides, and polysaccharides. We can compare, okay, between disaccharides and monosaccharides, and so you can compare, okay. And we shall use a gas pressure sensor. As we've said, a gas pressure sensor, it detects changes in carbon dioxide that is produced. So you are carrying out, this is anaerobic respiration, anaerobic fermentation. And in this, you are you are producing carbon dioxide, but you are not using any oxygen. And it's a closed system, it's a closed tube. Okay? So the carbon dioxide that will be produced, it will increase, okay, the pressure within this closed tube. Is that okay? And so if it increases the pressure within that closed tube, then the gas pressure sensor will be there to detect, okay, how pressure is increasing within that tube, within this closed tube. Whereby if you produce too much carbon dioxide, then there will be a lot of pressure that is detected. Okay? And the carbon dioxide is assumed to be coming from anaerobic fermentation of a given sugar. Okay? That's brilliant. If you produce less carbon dioxide, then there will be less pressure within this, and so the gas pressure sensor will detect less carbon dioxide. And so what does that mean? The sugar that we were using to metabolize was not a good metabolite. And so, based on that, we can get a curve of carbon dioxide respired vis-a-vis time. And you get such curves. You can see them. So amazing, so good. Okay? Whereby you can check on maltose. And then from those curves, I can say compare. Okay? Maltose vis-a-vis sucrose. Since they are all disaccharides, you can see. So I can say compare. Okay? The rates, the anaerobic fermentation of maltose and sucrose. And then from comparing, I say explain. Then I say compare. Okay? The anaerobic fermentation of glucose vis-a-vis fructose. And with comparing, you give differences and similarities. And as I said, we don't put differences in the table. For this case, for number 1, paper 2, don't put the differences in the table. But other cases, don't provide differences that are not in a table form. I hope I'm clear. I hope I'm okay. And from the graph, you can clearly tell that for lactose, together with its galactose, the pressure, the volume of carbon dioxide is constant. You can see there is no increase. Okay, it is constant. You see, for sucrose, it increases a bit, but finally remains constant. Okay, for this, it increases gradually from 0 to maybe around 40 or 35. Okay, now if you are attempting a paper, you have to be accurate. If it requires a ruler, use it. Okay, for those I told you, you know how I was strict with this. But if I haven't told you, then we shall get time and study graph work. This is where all, imagine one number with 40 marks. Okay, one number with 40 marks. So if you're not careful here, then you lose the marks. So you can see there is a gradual increase, and then from this to something like 125, there is a rapid increase, and then a gradual increase. So you should be in position to describe and explain. And from the above, you should be able to tell which sugar is best metabolized. And that's a gas pressure sensor. And we have another technique where instead of a gas pressure sensor, we can use methylene blue. And what is that methylene blue? This is an indicator that is blue in appearance, blue in color. Okay? It is blue in color. You know color blue. Okay? And we can use it to determine also which indicator has been, oh, which sugar is best metabolized. So how does it work? Now this methylene blue, it has two forms, the reduced form and the oxidized form. Okay? In oxidized form, this dye appears blue in color. In oxidized form, it appears blue in color. However, when it accepts electrons and it is reduced, then the blue color begins to fade, to fade, to fade, until when the whole of it is reduced and turns colors. And turns colors. Are you seeing this? Hmm? In oxidized state, it is blue in color. Let me repeat that. But however, when it accepts electrons, it begins to fade. The blue color begins to fade away, fade away, fade away, fade away, fade away, until when the whole of it is colorless. I hope that one is okay. Good. Now, you have blue. Okay? Where is my pen? You have blue. I hope this is blue. Okay? You have blue and you have white. So between blue, okay? Between blue and white, which one easily allows a beam of light to penetrate through? It may seem an obvious question, but that's where we are heading. So obviously, a colorless one, it allows full transmittance, 100% transmittance of light. And a deep blue solution, really, it is opaque. It doesn't allow any transmittance of light. What do I mean? If I have my solution, here the solution is blue, and I flash in light. If it is blue, I won't see any light at this end. But if the solution is colorless, then the whole of this light that is penetrating through this solution, it will be 100, the whole of it. The same way I propagated it is going to be the same way it is going to be transmitted through that solution. So that's why I'm saying you have a 100% transmittance. But here you have a 0% transmittance of light. But we have said that on accepting of electrons, okay, the methylene blue is reduced, and as it is reduced, it progressively turns slowly by slowly until it turns colorless. So as you bleach this, the blue color begins to fade away, so that if half of it is reduced, then it will have half transmittance, 50% transmittance. If 3/4 are reduced, then you have 70% transmittance. If a quarter is reduced, then we have 25% transmittance, and so on, and so on, and so on. Okay, we are moving on slowly by slowly. So the rate of dye change from blue to colorless in an indicator is an indication of the rate of breakdown of sugar to release electrons. So we saw that in anaerobic fermentation, okay, there is glycolysis, right? I hope that one is okay. And with glycolysis, you produce pyruvate, okay? And the pyruvic acid is converted to, let me change a pen, yellow can't be. the pyruvic acid is converted to ethanol. Okay, is converted to ethanol. Okay, with evolution of carbon dioxide. Now this carbon dioxide produced, okay, it is going to dissolve in the water, okay, H2O, to form that weak carbonic acid. Okay, carbonic acid. This is H2, and the carbonic acid will ionize to give you the hydrogen ions. Okay, now these hydrogen ions, they will be the ones to reduce the pH. Okay, they will be the ones to reduce the pH, and if you reduce the pH, then the methylene blue, okay, is going to be reduced. It is going to become decolorized. So basing on how much carbon dioxide you are producing, then it will determine how much you are decolorizing the methylene blue. Okay? Yes. So let us see. I think we have a question this side. So that is our first question. You can check. This was UNEB 2019. So if you were there, would you really pass it? I'm not going to answer it for you, but I want you to see what we are talking about. Sorry, what we are talking about. Sorry for that. Sorry. So, we have our methylene blue is an indicator which is blue when oxidized and colorless when reduced. The intensity of light passing through the mixture of solutions stroke reactants containing methylene blue can be measured. A 100% transmission of blue light means the methylene blue is completely oxidized. So here you are transmitting 100% of blue light. To be specific, it's blue light. Okay? Remember the other trichromatic theory in color perception of ours. Eh? The color that is observed is due to the color that is reflected. Okay? So if you reflect 100% blue light, then it means your dye is 100% blue, and so it is 100% oxidized. However, any deviation from 100 to 90, okay, to 80, now to 70 to 50, transmittance of blue light, okay, blue light shows that the dye is gradually degenerating, gradually decolorizing. Is that okay? And so now we have your three solutions. One has 10% yeast suspension, 10% of 10 centimeters cubed of 5% glucose plus methylene blue. Then another one, you have 10% yeast suspension, 10 centimeters cubed yeast suspension, 10 centimeters cubed of 0.5 sucrose plus methylene blue. So what is varying? It is sucrose. Here you have sucrose and here you have glucose. Then here you have 10 centimeters cubed this suspension, and then 10 centimeter lactose, and then methylene blue. So what is varying here? It is lactose. So here you have glucose, here you have sucrose, and here you have lactose. That's what is varying. And then the test tubes are covered at this water bar for 20. This one is just to provide the optimum temperature for the yeast enzymes for 20 minutes. 20 minutes is just to provide time, enough time for alcoholic fermentation. Then after 20, at 2 minutes interval, this is at every 2 minutes interval, the percentage of blue light transmitted through the test tube in each solution is measured, and then you get a graph. I told you this was UNEB 2019. Okay, and then the graph is here. So this is your graph. So this is the graph that you are getting. And then this is for lactose, you can see. This is for sucrose, you can see. And this one, you can see. This is a rapid and finally gradual for glucose. And then they start finding you questions. Compare glucose and lactose, sucrose and glucose. You can find that paper and answer the questions. Okay? Give a detailed account for the observed changes in lactose, sucrose, and glucose. Give it. You can see this one. It is lactose, and we said yeast does not have lactase enzymes. Glucose, it's a monosaccharide. This one is a disaccharide. So this one takes time. You have to first break down into glucose and fructose, and then you isomerase, and then you carry out glycolysis. You saw the whole process. But this is rapid. But it finally becomes gradual because you are carrying out within a closed system. So you expect the same carbon dioxide to alter the pH. The same alcohol is remaining within that system. So those are the wastes, and the wastes will inhibit the yeast cells from respiring. And really, I don't see anything big here, any big deal here. I don't see anything that should really become berserk on your side. Otherwise, if there are no questions, then this is going to be the end of our respiration. The graphs, they are there, the ones you've just had.